Okay, I'm going to go over the fundamentals of gel electrophoresis. Please forgive me for how bad these drawings are. I'm using a mouse, and I'm not good at it. Okay, so gel electrophoresis is essentially a tool that many labs are going to use in order to separate molecules based on one of three factors. One, and the most common, is size of the particle. Two is charge. And three is pH. So whether it's acidic or basic. So you can separate molecules if they have a different size, if they have a different charge, or if they have a different pH. And you can do different gels with different ingredients that will allow you to do sometimes multiple dimensions. We're going to first look at what's most common in DNA samples is size. When we put DNA in a gel, it's negatively charged. So DNA has a negative charge. And we're going to run an electrical charge through this gel in such a way that we have a negative pole on one side up here and a positive pole on the other. And negative particles are going to want to go to the positive pole. They're attracted to opposite charges and repelled by similar charges. So when we turn on the electricity and we put samples in up on this side, on the top of the gel, they should run down. Now this gel is literally a brick of gel. It's like hardened Jello. Okay, so it has a three-dimensional shape here. And in the shape, we put wells. So we take a plastic, what we call a comb, and we push it into the liquid when it's cooling and leave it in there until the gel solidifies. And we're left with basically a little container in here in which we can put a sample. So we put samples in here, and this is called a well. So that's one of our words to understand this technique, a well. And you put the sample in, and it's going to run in this direction. And this area where this, that should be a straight line, where this sample is running is called a lane. It's like a traffic lane. This is where the DNA is going to travel, or protein or RNA, whatever. All right. So on this gel, we have 6 wells, and if we put 6 samples in, we would have 6 lanes. We're just going to put in samples in the first 3 wells here. So we'll go ahead and put a sample in wells 1 through 4. When we turn the electricity on, the samples in here are going to go through the gel. And let's say we end up with a pattern on the gel. Okay, these little lines are called bands. And these bands represent samples of DNA. When you run DNA and separate something by size, the smaller the DNA fragment, the faster it's going to go through this gel. This gel is very thick, and at the molecular level, it's going to resemble a net, but let's not think about it that way. Let's just think about it as it's very viscous, very thick. And smaller things that have a smaller mass are going to be able to travel through this faster than things with a larger mass. So if I ask you, what is the smallest segment of DNA that appears on this particular gel, then you should say that it is the one that traveled furthest. So it is in lane 2, and it is this band down here that I've circled. If I ask you for the DNA that is the largest, just imagine that I drew this right and all 3 of these are on the same plane. They go straight across. And so it would be this one, and this one. And likely those 3 fragments are all the same DNA fragment. Okay, so they're the same lengths, and we're going to assume they're the same sequence of DNA. Now let's assume we're trying to determine the genotype of these 4 individuals. So we got samples of just one gene. We can do that. I'm not going to go into the technique. But we've gotten just one gene, and we have varying alleles for this gene. And we run this sample, and let's say it looks like this. Okay. And we've sequenced this gene and these alleles, so we know how long these alleles are. So in this case, these alleles have mutations that inserted or deleted some bases, so they're varying lengths. They code for the same gene, but they have different numbers of bases. And we're going to assume this is going from largest to smallest allele. It's just arbitrary, but that's what we're going to do. So allele 1 is the largest. So when we look at this gel, we can determine the genotype of these individuals. Here we have something that's very large compared to everything else. So this is probably allele 1. Okay. So let's do person 1, person 2, person 3, and person 4. And we want to know what are their genotypes. For this one, they have allele 1, so we know that. And they also have the smallest, and so we know that's allele 4. So this is their genotype, A1, A4. For this individual, they have the largest, so A1. And they have the second largest, so that's allele 2. So their genotype is A1, A2. The third person has allele 2. Just imagine it's at the same distance. I meant to, but just imagine it's on this plane. Just draw it there. They have allele 2, and they have allele 4. And the fourth person, which I've drawn it kind of over here, but the fourth person, their sample only has one band. It's in the middle of these two, so we can deduce that it's allele 3. So person 4 has allele 3, but what else do they have? Here's a situation where you just sort of have to use logic. If you have an allele 1, you're going to have a band for allele 1. If you have any other length of DNA, you'll have a second band. But if you have the second allele in your genotype is the same length, then you'll only have one band. They will not separate based on size because they're the same exact length. So in this case, it is safe to assume that this individual, their genotype is A3, A3. They are homozygous for allele 3. That's how you use and interpret this gel. Now we're going to apply it to proteins, which is the focus of the lab today. Today we're looking at the hemoglobin gene. And hemoglobin is a protein that is present in your red blood cells that is used to bind to oxygen and carbon dioxide. There are several alleles in the population, but the most common alleles are alleles HA, that means hemoglobin gene, and it is the version A. And then the second allele is hemoglobin with sickle cell. So there's two alleles that we're going to focus on. This is the normal gene, HA. This is the disordered allele, HS. These are also codominant. So remember, in a codominant situation, you don't have a dominant recessive relationship. If you have a heterozygote, both of these proteins are going to be expressed. So in other words, you will have two proteins instead of one. If you have HA, and then you're going to have the allele for HA, and you're going to produce a protein. We'll call this protein 1. If you have HS, you're going to produce a protein from that version of the gene, we'll call that protein 2. The sickle cell protein ends up creating a cell that is sickle-shaped because the protein is crystallizing in the cell. So now we're going to look at this gel. And in this gel that we're running and the questions you're going to be looking at regarding this particular experiment, we're running proteins. And so we're using the second property here, charge, to separate them. Proteins are going to vary in charge. They're not going to have a standard negative charge unless you do something special with them. So we're not going to do that because we want to see separation of these two proteins. And in this case, protein 1 is more negative than protein 2. And remember, this negative protein then is going to travel further in the gel than the positive protein. Let's look at some examples of genotypes and what proteins they would be producing. So in a homozygous individual for HA, these individuals are going to be producing protein 1, and that's all they're going to produce. They have 2 alleles, but they're just producing protein 1. The individual who has allele HA, HS, so this is their genotype, they're going to be producing the A protein, protein 1, and the S protein, protein 2. So they'll be in their red blood cells, in their blood, they'll have 2 different proteins. The homozygous for HS individuals will be producing only 1 protein because they only have 1 allele version. They have 2 copies of it, but they're only producing one protein. They're producing protein 2. Now we can look at these samples, 1, 2, and 3, over on the gel, and we can backtrack and say, okay, what was their genotype? And furthermore, we can, from their genotype, determine what their phenotype is. Do they have sickle cell anemia or do they not? Person 1, here we have a situation where we have 2 bands in this lane. So that means there's 2 proteins. One is more negative, that's traveled further, and one is more positive. So this individual having 2 proteins, automatically we know that person 1 is heterozygous for this gene. They're producing 2 different types of proteins. Person 2, they only have 1 band, and so we know they're homozygous, but we don't know what allele until we sort of use some logic to figure it out. Because this protein has not traveled very far, we know that it's the more positive protein. And so we can deduce that this person only has HS alleles. And so person number 2 is homozygous for HS. They have sickle cell disease. Person number 3 is homozygous for the negative one. So they are homozygous for HA. And that's how you run a gel, and that's how you analyze a gel. Remember, there's 2 different contexts here. We're using either DNA, which separates by size. So if you get a question about DNA, that's what's happening. Or if we're separating proteins, we're probably separating by charge. Okay, so 2 differences, and you should know how far particles are going to travel based on size or based on charge. For size, the smaller particle moves faster. For charge, the more negative particle will travel further.